You're listening to Innovating Tomorrow's Medicine with Dr. Jeff Trickett.
In this episode, I'm speaking with Courtney Young. With over a decade of experience in muscle disease research and therapy development, inspired by her cousin's Duchenne Muscular Dystrophy diagnosis, she has pioneered a therapy that aims to permanently correct a common mutation hotspot by editing the patient's own genome to restore a functional dystrophin protein in approximately 50% of patients. Currently founder and CEO of MyoGene Bio. Can you tell us about MyoGene Bio?
MyoGene, we're really interested in developing cutting-edge therapies for muscle diseases. So our CRISPR approach is basically turning Duchenne into this mild form of Becker to restore the protein that's otherwise lacking as a way to permanently edit the patient's own DNA, targeting the underlying cause of the disease.
So this can be used basically to edit a person's genome, their DNA?
Yes, basically it comes down to being a way to edit someone's own DNA, either in a cell or in the body.
So what are your biggest technical challenges you faced developing your CRISPR-based therapy?
Initially, we weren't sure how big of a deletion we could make. And then the second challenge is really delivery. It's still kind of an ongoing challenge for the field. And then, of course, safety is important. We want to make sure that our approach is as safe and efficacious as possible.
How do you handle the pressure and expectations that come with being a recognized young leader in biotech?
Being able to surround yourself with people who understand, who have done it before and can give you advice, or people who are going through the same thing you are, has been really helpful.
What advice would you give to other scientists looking to translate their research into therapies?
My main piece of advice is you have to do something you're passionate about. It's a hard journey to do something like this where you have a startup and you need something that you really care about and believe in order to overcome all the hurdles that you'll hit.
Courtney, welcome. Thanks for being here.
Thanks so much for having me.
Let's dive in. So what inspired your focus on Duchenne Muscular Dystrophy and gene editing as a therapeutic approach?
So I actually got into the field when my cousin was diagnosed with Duchenne when I was in high school. And his diagnosis was a shock to our family. We had never heard of this disease before, but it caused me to become really interested in working on muscular dystrophy. So I worked in a couple different labs working on muscle disease research. I worked on approaches with small molecules, with exon skipping, with stem cells. And I realized for my PhD thesis, I really wanted to do something that targets the underlying cause of disease in Duchenne. And I decided to go to UCLA for my PhD. And this was in 2013, right when CRISPR had just been published for use in human cells. And we said, maybe we'll try CRISPR for Duchenne. And that's what started my thesis project, which was developing a gene editing therapy for Duchenne Muscular Dystrophy, which we've now spun out into the company.
So you mentioned CRISPR, so aka clustered regularly interspaced short palindromic repeats. For the non-technical folks sort of like watching, I think we're going to flash up a graphic on the screen. Can you just sort of tell us a little bit about that approach and how it works?
Yeah, so CRISPR was originally in bacteria as part of an immune defense mechanism. It was a way to store pieces of phage DNA in the bacteria so that they could remember that DNA sequence and target that phage DNA when invading phages would come in. And scientists discovered this as a way to have programmable gene editing. It basically consists of two components. One component is a guide RNA, which you can think of as a flag. And when we use gene editing therapeutically, we can basically program this flag to go almost anywhere in the DNA. When that flag finds its target site, it brings along Cas9, which is a CRISPR-associated protein. And this protein acts as a molecular scissors. So it will go in, it will cut DNA. The traditional form is a double-stranded break in DNA. So your flag guides your molecular scissors. So it's basically a way to have programmable gene editing, cutting of DNA in a cell. And now people have adapted the Cas to be different forms. It can be a nickase, it can be a dead Cas, so it doesn't actually cut DNA. Instead, it can bring in other components, other proteins. So it's basically a way to have really site-specific targeting of either a double-stranded break or another protein brought into the DNA with this kind of two-component system.
So this can be used basically to edit a person's genome, their DNA?
Mm-hmm. So, traditionally it could be used to cut out a region of DNA. It could be used to disrupt a gene. It could be used to incorporate new DNA. That's with kind of the cutting ability. Or the other forms, it could be used to bring in another protein to turn on or off a gene, to cause base editing, to cause prime editing. There's a lot of different flavors of this. But yes, basically it comes down to being a way to edit someone's own DNA, either in a cell or in the body.
So can you tell us about MyoGene Bio, and can you explain how your lead compound, the MyoDis 4555, works and why targeting exons 45 to 55 is so impactful? And I think we, as part of that, we can also flash up your award-winning photograph of a muscle section that has been sort of like stained. So perhaps you can sort of tell us about that as well.
Yeah, so at MyoGene, we're really interested in developing cutting-edge therapies for muscle diseases. Our lead program is a gene editing therapy for Duchenne that uses CRISPR to make a deletion around exons 45 to 55 of the DMD gene. In Duchenne, patients usually have an out-of-frame mutation. This is a change in the DNA that basically results in the puzzle pieces of DNA no longer fitting together. And that will make the gene non-functional, and no dystrophin protein will be produced, which normally protects muscle. And as a way to kind of restore the reading frame so that a protein is able to be produced, we've developed a CRISPR therapy where we use guide RNAs, we use those flags to target intronic sequences around exons 45 to 55. And that will cause Cas9 to go in, make that double-stranded break, and delete exons 45 to 55 of the DMD gene. This then makes an in-frame deletion. That means those puzzle pieces of DNA will fit together again so that an internally deleted but still functional dystrophin protein can be produced, and this is expected to significantly improve function in Duchenne. We target exons 45 to 55 for two main reasons. One is that it encompasses a hotspot. Around half of all patients have mutations within these exons. So with one approach, we can target a large proportion of patients. And then secondly, the resulting deletion, lacking exons 45 to 55, has been associated with a very mild form of an allelic disease called Becker muscular dystrophy. So some Becker patients with a naturally occurring exon 45 to 55 deletion have a very mild phenotype. Some have been asymptomatic until their 60s, whereas in Duchenne, patients typically die in their 20s or 30s. So our CRISPR approach is basically turning Duchenne into this mild form of Becker to restore the protein that's otherwise lacking, as a way to permanently edit the patient's own DNA, targeting the underlying cause of the disease.
And can you explain about the photo and muscle staining? Could flash that up?
Yeah, so some of the work that we've been doing has been using AAV to deliver CRISPR in vivo. So we can inject AAV carrying CRISPR into the bloodstream of mice, and it will go into the muscle and deliver the cargo. So we demonstrated that AAV can effectively deliver proteins to muscle by delivering GFP and mCherry. So in the immunostaining picture, when you see the filled-in muscle of either green or red, that's delivering those GFP and mCherry proteins. And then we've shown that we can use AAV to deliver CRISPR itself, and this will restore dystrophin to the protein, to the cell, and this protein then is able to improve the function of those mice.
And then in terms of restoring the dystrophin, and again, as you know, I used to work in the Duchenne field some time ago now, but this is not just restoring dystrophin to skeletal muscle, but also other sort of tissues that are affected, so the cardiac muscle and also diaphragm muscle.
Yep, exactly. So because we give a systemic injection, we're able to target multiple muscles in the body, including heart and diaphragm, which are very critical for a patient's function.
Yeah, very, very, very neat. So what are your biggest technical challenges you face developing your CRISPR-based therapy?
Yeah, so initially we weren't sure how big of a deletion we could make. So the deletion we want to make around exons 45 to 55, we target the outside edges of the flanking introns. And a wild-type gene is actually 708 kb. It's a really, really big deletion. And so the first work we did was testing different patient mutations within this region to see what size deletions we could make efficiently. And we actually showed we could delete up to 725 kb efficiently. So in a patient that had a duplication, for example, we could still delete this region efficiently with CRISPR. So that's really demonstrating the power of gene editing, is you can make really large modifications in the DNA. And then the second challenge is really delivery. It's still kind of an ongoing challenge for the field. We tried a variety of non-viral approaches for muscle, but we found AAV to be the most efficient delivery for systemic injection to muscle. So that's what we're currently advancing, but we're hopeful that innovative delivery approaches will still continue as a way to have a non-viral approach for delivery to muscle. And then, of course, safety is important. We want to make sure that our approach is as safe and efficacious as possible.
And then in terms of the stratifying your patients, so just your starting point is you're trying to sort of like cure sort of 50% of patients, but this technology has the potential to sort of be used for if not all, then certainly the sort of greater majority. I mean, dystrophin is, I think it's the largest human gene, so there's just by definition there's more of it to go wrong, but there's sort of point mutations, there's deletions, all sorts of things. But I think the potential is there to treat the other sort of like variations in the gene.
Yeah, definitely. One of the advantages of CRISPR is it's not dependent on the type of mutation the patient has. It's really more dependent on the location of the gene that you're targeting. So when we target exons 45 to 55, it works for any type of mutation within that region as long as the guide RNA target sites are present. And we plan to kind of use it as a platform approach where we can take our strategy that we've used here and apply it to other regions of the DMD gene to encompass more patients. In Duchenne, there's unfortunately not another single hotspot of another 50% of patients. You kind of have to piecemeal the other 50% of patients, but I definitely think there's potential to get a large number of patients using an approach like this.
Very neat. So how does your therapy differ from other gene therapies or editing approaches currently in development for Duchenne muscular dystrophy?
Yeah, we think of gene editing as the next generation of gene therapy. There are some gene therapies in the clinic and one approved for Duchenne already. But gene editing has some advantages. You're permanently editing the patient's own DNA, so that has potential to be longer lasting. It keeps the normal gene regulatory environment intact, so it's the same promoter as the natural gene. You don't incorporate any new DNA sequences, and we're not delivering a replacement of the gene, we're delivering CRISPR. And so that allows us to make a much larger version of the dystrophin protein than is possible with the gene replacement therapies. So when you use AAV to try to deliver a copy of the dystrophin gene, it's so large it doesn't fit. And so people have made mini and micro versions of that gene. These are about a third the size of normal, whereas our edited dystrophin is about 90% the size of normal. So we're able to make a much larger protein encompassing additional functional pieces, aiming to be more effective. Compared to other editing approaches for Duchenne, the main advantages of our target are that we encompass 50% of all patient mutations with one approach. If you just target one exon, it's only 13% of patients or less. And we're using CRISPR, which is an already approved technology that has already been FDA approved for other diseases.
What preclinical results have been most encouraging so far?
Yeah, we've done a lot of work of our efficacy studies in human cells and a humanized mouse model of Duchenne. So in the humanized mouse model, we can give a single systemic injection of AAV-carrying MyoDys 4555, and we've showed that this can restore dystrophin to levels that we predict to be functional, 15 to 30% total protein, in heart, skeletal muscle, and diaphragm. And then when we look at the function of these mice, we do see a significantly improved function, basically to a level of the healthy control mice. So I think that's really exciting that we can really target the underlying cause of disease in a humanized mouse model, restore the protein, and improve their muscle function.
What are the major regulatory hurdles ahead of you as you move towards clinical trials?
Yeah, so the work we have to do to get to trials include kind of the IND-enabling safety work, so showing that the approach is safe in a large animal model, work on manufacturing scale-up, and then the clinical trial design. Because we're working on a rare disease, we can take advantage of some clinical trial incentives, but we still need to make sure we're measuring something that's impactful and looking at something that means something to the patients.
How do you envisage the future of gene editing evolving beyond DMD treatment? Would be good to bring out the personalized medicine sort of like approach here.
Yeah, so gene editing is being used in a lot of different diseases, not just Duchenne. It's been used ex vivo and in vivo, both preclinically and clinically. And it's shown a lot of power for being able to make genetic changes. These approaches are either editing DNA or RNA, they're either making double-stranded breaks or doing things like base editing or gene activation or inactivation. And so there's a really wide potential of what CRISPR can do on the DNA level. The ability to use it for personalized medicine has also been demonstrated. There was a recent case example of a baby that got a personalized CRISPR therapy developed within 6 months, and they were able to treat that baby that otherwise, would have suffered a severe disease. So I think the potential of CRISPR is that you're able to really specifically target DNA, and that could be done on an individualized basis.
Moving on to leadership and more about the company. So what lessons from your PhD and postdoctoral experiences have been most valuable in running a biotech startup?
Yeah, I think running the project is kind of similar to what you do in academia, although your goals are different. So in my PhD, I was fortunate to have a variety of people help on my project. My, advisors were supportive of the work and, had some people help me. And so I had to run my thesis project kind of like how you would run a company in a mini format. So I think, being able to organize people and run a project was a really good learning. And then some of the biggest things you learn in PhD and postdoc experiences about, critical thinking, problem solving, and deep scientific and technical knowledge. And since we spun out my PhD thesis work into the company, it's very clear that I have, the scientific and technical basis for that work as we work for in the company. So it was a really good learning experience, and then also, bringing the actual scientific knowledge into the company.
And that neatly leads to the next question. So how do you balance the roles of scientist, CEO, and advocate?
Yeah, there's, a lot you have to do running a company, and I'm also basically the CSO at the time. So I do have to balance the kind of science roles, the advisory roles, the executive roles, and then some administrative work also. So I think I kind of split my time depending on what the most urgent needs are of the company, but I'm very passionate about running the science, and so I do that with a good amount of my time. Lots of fundraising efforts as the CEO type role, and then kind of the administrative work of running the company. And then always, trying to connect to the community through those roles. I obviously have, connections through my family, and then attending different conferences and networking with people is, still very important kind of for the advocate side.
What has been the most rewarding part of leading MyoGene Bio so far?
I'm really impressed with the team that we have and the progress we've made on a small amount of funding, staying very lean, but having people really dedicated to the company, where we're now advanced enough that we are beginning to have a line of sight to clinic, which is really the overall goal of the company, is to, get something to patients. And we're finally at a point where we're beginning to see that happening. And so I've been, really excited to see that, and really proud of the team that has got us here. And we've gotten a lot of non-dilutive grants, participated in pitch competitions and things like that. And that's all really validating also from the community to show that, other people are believing in us also.
What's the biggest challenge you've overcame when making the transition from academia to industry?
Yeah, when we were spinning out MyoGene, the biggest challenge we had was kind of learning the language and how you tell your story in the business VC world rather than academia. In academia, you want to tell your story all about science, but in business, you have to make the business case. And so that transition, and even just knowing all the acronyms and all the phrases that people talk about in the business world, I think was kind of the initial learning curve we had to overcome. But in grad school, I took some boot camps. I took a couple of business classes, a summer program. So I had tried to build up some of that, but ultimately when you're doing it is when you really learn how to do that. And it's very different than in academia.
What partnerships or collaborations have been key to your progress?
Yeah, the academic research collaborations we have are really important for some of our efficacy work. There's animal models that we need to be able to use that are applicable for our disease and our target. Also for manufacturing, having an experienced manufacturing partner is important. And then ultimately for the clinical trials, we'll also be looking at partnerships.
How do you handle the pressure and expectations that come with being a recognized young leader in biotech?
Yeah, the San Diego environment has been really supportive. There's a lot of startup companies here. I'm in some CEO networking mentorship groups where you have peer mentorship. And so being able to surround yourself with people who understand, who have done it before and can give you advice, or people who are going through the same thing you are, has been really helpful. It's kind of a lonely job being a CEO. And so the more you can network and have, relationships with people who understand is really helpful. So I'm very appreciative of everyone around me who has supported me, and also, peers who are going through the same thing that we can all commiserate together.
So I now have the previous guest question, just a bit of fun, and I think it's an example of that we take our work very seriously, but we don't always take ourselves seriously. So with that said, the question is sock, sock, shoe, shoe, or sock, shoe, sock, shoe?
Sock, sock, shoe, shoe. Definitely. Yeah.
Yeah, it's a big debate online right now, but I'm definitely a sock she person. I've always done it like that. But anyway, thanks to our previous guest for that. And moving on to sort of like vision and impact. So, what impact do you hope MyoGene Bio's therapies will have on patients and the rare disease community?
Yeah, obviously our main goal is to develop truly meaningful therapies for the patients. We want something that can really change the trajectory of disease in Duchenne and then other potential muscle diseases that we hope to target. These are diseases that really have no cure, and these patients will die if they don't have new therapies. So we're hopeful that gene editing can offer a solution where you can really target the underlying cause of disease to have a really meaningful impact on a disease progression. And I think the power of gene editing and gene therapies are really at that point where we're beginning to see, you can really kind of have true impact on patients' lives.
How important is patient engagement and feedback in shaping your research and development?
Yeah, we're obviously doing this for patients, so it is important that we listen to the patient voice. We actually have a grant right now where we have a panel of patients and patient advocates and other people in the community, as well as advisors, both scientific and clinical, so that we can get, direct patient feedback on our program in a structured manner. We go to conferences, we interact with the community, but it's also really nice to have kind of these structured interactions where you can get feedback from people, because ultimately you are doing this for the patients.
What advice would you give to other scientists looking to translate their research into therapies?
My main piece of advice is you have to do something you're passionate about. It's a hard journey to do something like this where you have a startup and you need something that you really care about and believe in order to overcome all the hurdles that you'll hit. So it's really important that it's something that you're truly passionate about. In this day and age, I would also say be creative with fundraising. It's a challenging time right now. So the more you can be creative with funding and staying lean, the better you'll be off in this environment.
And how do you see the biotech industry changing in the next 5 to 10 years, especially regarding rare diseases?
Yeah, with rare disease, I'm hopeful we'll see the idea of platforms being brought out. So individually, rare diseases are very rare, but all rare diseases together are very common. There's, a large number of people with rare disease. And I think, there's been a lack of pharma interest in some of the ultra rare diseases because of a lack of market opportunity. But if there's a platform approach where you can take a similar type of gene therapy or gene editing therapy and target multiple rare diseases, all of a sudden that becomes a more attractive market opportunity. And so I'm hopeful that the idea of platforms could become more common and make rare diseases more attractive for pharma, which ultimately will benefit patients by allowing, really meaningful therapies to be brought forward.
In what ways has inclusive leadership directly contributed to scientific or business innovation in your organization?
Yeah, so MyoGene is a fully woman-founded and run startup. So from our core, we're very passionate about inclusive leadership and diversity. I think having a diversity of perspectives is really critical when you're running a company and working on an innovative therapy. You want people from different backgrounds and different experiences. You don't want someone who, knows the same thing as someone else, because, you're kind of then wasting space in your company. So being able to have a variety of perspectives, diverse experience, and being inclusive is really critical.
And coming back to the funding, how have you funded MyoGene Bio to date? And again, especially given it's been a more challenging environment at the moment.
Yeah, the majority of our funding has been non-dilutive grants. This includes from the NIH, the DoD, the Muscular Dystrophy Association, and the California Institute of Regenerative Medicine. And that's really beneficial because you can then leverage this non-dilutive funding and progress the research. We've also had some venture philanthropy investments and angel investment support from Duchenne foundations, which are really validating support from patient communities. And that funding, can supplement our non-dilutive grants. So the majority of our funding has been from grants, but the, venture philanthropy and patient foundation funding is also very valuable.
Courtney, thank you very much.
Thank you so much for having me.
Innovating Tomorrow's Medicine with Dr. Jeff Trickett.